Thank you. Welcome, everybody, to a very special Pediatric Grand Rounds today. We have a special guest, and I'm going to be actually turning this over to Brian Feldman, who's our inaugural Walter Miller Endowed Chair, to get us started and introduce our topic for today. So, Brian.
All right, well, welcome everybody. I'll take my mask off so you can hear me. But I hope everybody is at least half as excited as I am. It's been a long road that was detoured by COVID, but welcome everybody in person and those of you on Zoom. We hope to get more and more people here, and that this will be the first year of many that will have this inaugural, have this Walter Miller Lectureship. So first, I want to thank Dr. Hirsch for allowing us to start this lectureship, as well as thank some people that really worked hard behind the scenes. Susan Antalon, Miriam Farshad, Dylan Oren, and the development office all were instrumental in making today happen. And I also want to thank our generous sponsors, in particular ArborMed. So as you may or may not know, ArborMed is an early stage pharmaceutical company, but they have no drugs on the market, so they're not here to sell you anything right now. But they're a really good partner because they indeed are very interested in early stage and opportunities to interact with academics that are thinking about trying to translate their work. And so I think they really share many of our interests about basic science and how it can lead to new therapeutics. And our other sponsor, Corcept, which has been around for a little while, but it's still on the relatively small side, but interested really in novel aspects of steroid hormone signaling and unique ways to use our molecular insights about hormonal signals to develop, again, novel therapeutic approaches for diseases that don't have good therapies already. So you may be wondering why I thought we needed to have a new lectureship. There are already many established here, but I felt like there was actually a need to highlight the physician scientists a little bit more at the university. And I thought this was a really nice opportunity to do so. And in particular, I think that those of us that work on the basic science side of the spectrum really are able to see how basic science plays a role in actually the care and health of humans. It's not always obvious early in training that how that happens. And so I thought a really great way to illustrate that would be to bring people to the university that have really made instrumental, fundamental contributions from that angle, from that perspective, to demonstrate how important the physician scientist's role is, particularly the more basic side that you may not be exposed to previously. And so that's really the goal of this lectureship. And I think our inaugural speaker really demonstrates that quite nicely as you will learn from. But I also want to now first introduce Dr. Miller and explain to you how we came to decide to name this lectureship after Dr. Miller. Many of you know Dr. Miller from his many years here at UCSF. So I'll just keep this very short and then allow Dr. Miller to introduce Joel. So Dr. Miller actually went to Duke for medical school and then spent some time at MGH for his internship and residency, and then came to UCSF actually as a senior resident after a slight detour at the NIH in the division of endocrinology there. But he's been at UCSF ever since, rising through the ranks all the way to the top to become a distinguished professor here at UCSF in 2005, and then becoming emeritus in 2014 after serving as division chief of pediatric endocrinology for about 15 years. So his research is really widely appreciated as being some of the most fundamental in terms of our understanding of actually a broad array of aspects of pediatric endocrinology, but in particular, the adrenal gland and its steroidogenesis. And his work has actually identified previously unrecognized diseases. And I think it exemplifies how understanding the molecular and basic science behind pathogenesis of disease can lead to new insights and development of therapies with wide-ranging implications for human health, including the children that we care for. In addition, Walt, I think, has always remained true to himself. In particular, is known widely as a rigorous scientist and a defender of a meritocracy that we all aspire to maintain here in academics. So I think that these are all aspects of physician scientists that we all want to include in our own thoughts and make this worthy, make him very worthy of having a named lectureship in his honor. So I'm now going to ask Walt and Joel to come up here so we can present him with the plaque to kind of commemorate this nice occasion. But I do want to just mention as he comes up that subsequent to this talk, we will be having a reception where we invite you to join us in the atrium area of the SF Kebab, and we'll march over as a parade to make sure nobody gets left behind. But Joel, congratulations. We welcome you and thank you, and want to, as an expression of our gratitude, present you with this plaque.
Thank you.
So now you get to just stand there and listen to me bloviate for 2 minutes.
My pleasure.
Okay, so first I wanted to thank Brian Feldman for that warm introduction, and especially for creating and organizing this event. And also wanted to thank ArborMed and Corcept, which he has already mentioned. So it gives me great pleasure to introduce Joel Hirschhorn, whom I've known for a long time as the first Miller Lecturer. Joel is one of America's foremost pediatric endocrinologists. He's the chief of pediatric endocrinology at Boston Children's Hospital and is professor and Concordia professor of pediatrics and genetics, both at Boston Children's Hospital and the Harvard Medical School. And he's a member of the Broad Institute at MIT and Harvard. His research focuses on genetic variations that influence diseases and quantitative traits. He chairs the GIANT Consortium, which has discovered many of the variants associated with obesity and height, and recently mapped nearly all of the human genomic loci that influence height. His lab also studies diabetic kidney disease, Ehlers-Danlos syndrome, disorders of sexual development, relations among diet metabolic levels, and cardiometabolic disease. In other words, pretty much all of pediatric endocrinology and then some. His lab mainly utilizes human genomics, but he also develops computational and experimental methods to integrate genetics, metabolomics, and other genome-wide data. He also continues in his copious free time to see pediatric endocrine patients, both in the hospital and in clinic, all while running a large division that is academically and clinically superb. Joel received his baccalaureate degree summa cum laude in biochemistry in 1986 and his MD PhD in 1995, both from Harvard. He did his pediatric residency and pediatric endocrine fellowship at Boston Children's Hospital, and then did a postdoc with Eric Lander at the Whitehead Institute. He has directed the metabolism program at the Broad Institute since its inception in 2004, has helped to direct the human genetics course for first-year med students at Harvard since 2006. Joel was the recipient of the Mead Johnson Award from the Society for Pediatric Research, was then elected its president, and has recently been elected a member of the National Academy of Medicine. So I am really honored to have such an outstanding pediatric endocrinologist deliver the first Miller Lecture.
Well, I view this as quite an honor. This is, I haven't done yet too many in-person lectures, but this is one that I felt, privileged and compelled to accept. To be able to participate in honoring Walt Miller. When I was a child, I read Gulliver's Travels, and for those of you who haven't read it, it's a satire by Jonathan Swift. But one of the lands that that Gulliver visits is Brobdingnag, which is sort of the land of the giants. And that was sort of my conception of UCSF pediatric endocrinology. May not maybe everybody wasn't sort of a physical giant, if you remember sort of who was around, but it really was, you know, I mean, it remains a superb place, but Walt was, you know, part of that sort of crew of giants that was just amazing to be able to interact with. And I think there's sort of a couple of aspects of Walt's sort of career and approach that that I want to highlight. So one is that I've appreciated that a lot of the discoveries have come from studies of patients, and even, you know, studies that the genetics has really kind of helped reveal, you know, what's going on, and given sort of the, you know, clues to the biology, but then sort of the ability to take that and, you know, turn that into physiological insight and therapeutic leads, and just sort of do that. I actually have a slide, and, you know, sort of unusual to find somebody who's sort of made it all the way around the circle, you know, of genetics to insight to and to keep going. The other thing that I have to say is that, you know, Walt's often able to see things that maybe are not quite as apparent to other people. So, you know, that's not just when it comes to, you know, discovering new pathways for adrenal biosynthesis, but I remember one of the last times we had an extended conversation, or maybe it was 2 times ago, I think actually, Kyrie Irving was had just started with the Celtics, and I was sort of mesmerized by his ball handling ability, and, you know, it was like, oh, how much fun he was to watch. And I remember you saying, he's a ball hog, he's going to wreck your team. And you were totally right. You know, so, and it even pains me in San Francisco to bring up the Celtics, and, you know, being so close to the scene of the crime from last year. But yeah. But anyway, so, you know, you've sort of in many different ways had that sort of, you know, insight and you know, courage of your convictions, even ahead of what other people might have seen. So it's just a real privilege and honor to be able to be the first of the Miller lecturers. So thank you also for the kind introduction, and I'm going to jump in now, trying to tell you about the work that I and many people as part of the GIANT consortium, most of the work that I'll talk about is under the rubric of GIANT, have been doing to try to understand the genetic basis of various endocrinological diseases and traits, with a sort of focus on human height and measures of obesity. So these are my disclosures, which don't have, other than, well, data from 23andMe will have an impact on the talk, but no financial relationship with them. So why is human genetics a good thing to study? At least in my view, there's 2 reasons. The one which got me into it is that when you have genetic variation that influences a disease or a quantitative trait, and I'll highlight this more as the talk goes on, you know that there's a causal relationship there. It may be a complicated causal relationship, but it tells you this is one of the root causes, and it's up to you to figure out the clues that genetics has left for you. And that's kind of a theme that'll run through the talk. There is, of course, also the way that many people think about the role of genetics, which is in the context of monogenic disorder. It can also, especially, although perhaps now polygenic disorders, it can provide diagnosis or predictive information to patients and a much more immediate impact. But I'm going to spend most of the talk talking about sort of the idea of using genetics to understand biology, human biology. So here's this circle that I was alluding to, where, you know, in my view, you know, if you want to understand what you should do to relieve human suffering, you have to understand the disease in humans in some way or another. You know, there are certainly useful models that are non-human models, but, you know, the human is a good model organism if you can figure out ways of studying them. And so one of the ways is actually to use genetics, where you have sort of nature's experiments of altering the genetic code that they've inherited, or altering the genetic variants that they've inherited, and then trying to figure out what sorts of biological insights you can glean from understanding the impacts of those genetic variants. So why would genetics help us understand disease? Well, some of that you can get from thinking about how much variability that you observe in the human population is due to inherited factors. So, you know, if you have identical twins, not only do they, you know, pretty often look alike, have the same height, more or less the same weight, but they have many similarities in disease risk, as opposed to when you sort of look at, a random collection of people. This isn't a random collection. This is the X-Men cast. But you can see there's much more variability. These people are not identical twins, or even I think any of them brothers or sisters. But you can see there's much more variability in height, weight, lots of other things that you can see, and also their disease risks will be much more divergent from each other than for identical twins. And so a lot of this, although not all of it, because there's also more difference in childhood environment, for example, with a lot more genetic variability comes more phenotypic variability, and that the amount of that correlation is called heritability. And that's true for most of the diseases that you see in the hospital, and sort of most of the quantitative traits that you can measure in people. And so what is the genetic variability that's contributing to this? So we think that most of if you compare any two people in this picture, or any two genomes, even my genomes, one from my mom and one from my dad, you'll see there's about 99.9% identity, or 0.1%, or 1 in 1,000 differences. And we think that most of those differences have no effect, but some of them do have phenotypic consequences. And but generally, we think, at least particularly for the ones that are common in the population, so that you might find them somewhere in the room, or somewhere in somebody watching by Zoom, those effects are typically modest. And so most of the differences that you see in this picture, the genetic contribution to those differences is usually due to the combined effects of many different genes. That's not always true, though. So here's Peter Dinklage, who's a really great actor, who, according to Wikipedia, and seems likely, has achondroplasia. And so there you have a single base change that has a very large effect, in this case about 16 inches on average of height. So, but again, we think that most variability, and so most diseases, most traits, are actually what are called polygenic. So they actually, and this is another science fiction cast for those who are old enough to remember them, where you have many different genes in any individual person. So you might have the high risk version of gene 1 and the low risk version of gene 2, the high risk version of gene 3, and so forth. And probably in a largely additive, we think, way, the risk or the contribution to quantitative traits for each of these genes kind of adds up and determines the total genetic risk for that particular phenotype. And of course, there are also non-genetic factors, which could be environmental or random chance or other things as well. So we study the genetics of height for a number of different reasons. One is it is the classical polygenic trait. The idea of a polygenic trait was actually more or less invented or described by Fisher in 1918, talking about the variability in height and how you could get sort of a normal distribution based on contributions from lots of different genetic factors that all add up. But it's been used for hundreds of years. In fact, genetics of height's been studied even before genetics was a word. And so very basic concepts like regression analysis or correlation coefficients or things like that actually come from people who were trying to invent methods to understand the genetics or the phenotypic variability of height. It also is a very useful model trait in another way, because there's been a lot of work done to study the monogenic causes of short and tall stature. There are now hundreds of these genes that have been identified, and we call these in the talk and in our papers OMIM genes, because that's where they're cataloged in the online Mendelian Inheritance in Man catalog. It's highly heritable, so within any given population, the fraction of variability that is due to genetic factors is typically 80% or higher. It's routinely and easily measured. So in any genetic study, you typically can also do a genetic study of height. And of course, a big reason why I got into it is obvious relevance to pediatric endocrinology, because one of the more common referral that we get is for short stature. All right, so if you look at the distribution of height, like I said, it's pretty normally distributed. There's actually a little bit of a thick tail on the short end. And most of that variability is due to these small effects of lots of different common variants. But again, particularly at the short end, although a little bit at the tall end too, there are individuals who have monogenic causes, in this case depicted of short stature. And the reason that I sort of go into that is that the way that you go about trying to find these genetic variants, either the common ones of modest effect or the ones of large effect, which tend to be quite rare, depends. So for the common polygenic ones, you use a method called genome-wide association studies, which I'll talk quite a bit about. But for the rare monogenic ones, there's sort of family studies or large sequencing studies, which I'll allude to very briefly. Okay, so I'm going to talk a little bit about genome-wide association studies, and this is just to illustrate how you would do this for height. So the first step is to get DNA from study participants and then analyze it at sites that are already known to vary commonly in the population. So if there's, for example, a site where there's some people have a G and some people have an A, you'd find both versions at sort of, let's say, 1% of chromosomes or more. You then measure people's heights, and then you collect genetic data at those sites of variation and determine what genotype people have at each site. So is it 2 Gs, 2 As, or 1 G and 1 A, one from each parent? And so then you go systematically through each of the variants and you look for an effect. And in this case, there's no effect. And in this case, greatly exaggerated, because if we found effects like this, we would have been done with this whole exercise a long time ago. But in this case, you can see that there's an effect of the genotype on height, and that's what you're looking for. And so people have done this for height and many, many different other traits. And so this is the sort of 2022 version of this map. Each of the little groups of little tiny colored circles is a different chromosome, and each little circle is a place on that chromosome where there's been a robust association between a human phenotype and genetic variation at that spot, and the different colors are different types of phenotypes. And the upshot of that is that there are hundreds of thousands of genetic variants that now are reproducibly known to affect human biology and disease. And thus far, because of the methodology largely that's been used, but perhaps a little bit due to the underlying genetic architectures, most of the variants that are on that map are relatively common. So things with allele frequencies 1 to 5% or more. And as I said, the impacts of these variants are small, but as I'll hope to at least briefly convince you, the potential impact on biological discovery can be big. So one piece of evidence for that is that even early on, a lot of the, if you looked where the variants showing association with different traits or diseases were, and you looked at the genes nearby, you could find the sites of action of known therapies. And that includes for height, pretty much all of the kind of known or in trial therapies that there are. So the hope is that if you find new genes that you can convince yourself are relevant to the phenotype from genetic evidence, that those might be new therapeutic leads. And in fact, this is sort of the first of these types of papers, and there have been follow-up papers as well, showing that if you have genetic support for a gene being a good target, and that could be either from monogenic or from the genome-wide association studies, the likelihood of succeeding or failing at the efficacy stage was influenced by kind of 2 or 3-fold. So your study is kind of 2 or 3-fold more likely to succeed if you have underlying genetic support. All right, and so I made this claim that even though the effect sizes can be small, the importance could be large. And I think the reason for this is that most of the variants that we've been looking for are common. And so by definition, those with very rare exceptions, those have flown under the evolutionary radar. So if something has a big effect on physiology, regardless even of whether it affects disease outcomes, then evolution will notice typically, and it will either get amplified and sort of fixed in the population, or it will get prevented from becoming common. It won't sort of live in that limbo of being a common genetic variant where both alleles are tolerated. So for these common variants, the effects on biology, almost by definition, have to be limited with, again, some unusual exceptions. So that doesn't preclude the fact that if you have larger biological effects, there might be more profound effects on outcome. And so that's even illustrated by genetics. If you have a common variant with a sort of small impact on gene function, that might have a small effect on outcome. But we know that when you have a monogenic rare variant of large effect, that can have a huge effect on gene function, and then a similarly huge effect on outcome, causing a monogenic disease. And the idea is that pharmacological interventions are not limited by this sort of evolutionary radar. They can have big perturbational effects on biological pathways. So they could have big effects on outcome, even though they're affecting the same gene. Now, this does tell you that it's part of the causal path. So it can provide a very useful biological clue. But as again, I'll sort of point out, there could be many steps and many difficulties in figuring out, first of all, what the right gene is, but then also what the connection is between that gene and the outcome that you're interested in. And understanding that is often key to understanding where the therapeutic lead should be. All right, so we set out to try to understand the genetics of anthropometric traits, so height and measures of obesity. And knowing that the effect sizes already were going to be small, we knew that we were going to need large sample sizes to detect them at robust, rigorous levels of statistical significance. So we formed a consortium called GIANT, and in the sort of about a decade ago period, these were the initial set of cohorts that were participating. And actually QCing all of that data from lots of different people turns out to be a large effort. And so this is the group that was doing kind of the data cleaning and putting it together. And the effort for height was led by Salaja, Andy, and Tonu for the data that I'm about to show you. So this was the results of our studies in 2014. This is what's called a Manhattan plot. You can imagine sort of skyscrapers. And on the x-axis are each of the sites where there's common genetic variation laid out in order of chromosome across the genome. And then the y-axis is the strength of association with height. Anything above that dashed red line is robust association. So there are lots of these skyscrapers that cross that threshold. And in fact, there are 697 in this, it turns out. And if you look near those signals of association, it's evidence that we're on the right track because they're strongly enriched for these OMIM genes that affect short stature. And at this point, we were able to explain about 10% of height variation, which it turns out, although we didn't know it at the time, is about a fifth of the variation that you can expect to be attributable to common variant genetics. So about half of height variation is attributable to common genetic variation. So we've now gone on to do a much larger study. This is about 20 times the sample size. This is actually just out yesterday. This is, first of all, more diverse than we've done in the past, although still, unfortunately, predominantly European ancestry individuals, which is still a major limitation of the field, not just for height, but for every other genome-wide association study pretty much that's been done. But we still were able to get a total of about 5 million individuals, and the group that sort of led this in various different ways is shown there on the right. Particularly Loic Yengo. And for any genetic GWAS aficionados in the audience, there are some methods boxes. The rest of you can safely ignore those. But essentially what we found was that there were about 12,000 places that independently were associated with height when we put the data together for all of the different ancestries. And when we look nearby, it's still enriched for these OMIM genes. So we're still on the right track, although there's some interesting subtleties about where the variants are related to those genes as you get sort of lower and lower down in the effect size spectrum. The other thing that we found was that if you looked at where those 12,000 variants were, they were not sort of spread evenly across the genome, but actually they clustered in about a quarter of the genome. And it turns out, using various methods, that you can estimate that almost all of the common variant heritability lies in this quarter of the genome. And furthermore, when we take these 12,000 variants and try to see how much of height variation can we predict, it's about 40% just from those 12,000 variants. And if we dig a little deeper down into the rest of the genome, you can actually get about 45% of variability. And again, that's compared to the 50% kind of max threshold that we think we can get. And this is in European ancestry samples because of the way we did our discovery and because of differences in correlation between variants in Europeans as opposed to non-Europeans, particularly those with African ancestry. We do less well in predicting height for non-European ancestry. But we think we've basically, at least for European ancestry, gotten most, the great majority of the variation that contributes to height. And, certainly saying that this, this goes beyond, for example, what you could get for mid-parental height. This actually explains about a third of the variability in siblings who obviously all have the same mid-parental height as each other. And when you put it together with mid-parental height, again, in European ancestry folks, you now get about 55% of the variability in height. All right, so and I'll get back to sort of, potential use of those predictors at the end. But like I had said at the beginning, we're interested in how do you go from these genome-wide association study associations, these associations with common variants, to understanding the biology. And it turns out that if you zoom in on a particular region for a Manhattan plot, you can see some of the difficulties start to emerge, which is that you have, if you look, for example, on the right here, here we go. There's a cluster of variants here, and they're color-coded by how tightly correlated they are with each other. And you can see that it's hard to tell which of these variants is actually the causal one because they all have almost equivalent genetic evidence. And furthermore, a lot of them are non-coding. And if you then look down at the genes here, oftentimes, particularly, even for height where we know a lot, there's no gene that sort of makes obvious sense. You can't say, oh, well, of course, it's, that's the right gene. And because these associations are largely non-coding and not even correlated with a coding variant, it's also that makes it hard to know which of those genes is implicated because regulatory variation can act at a distance. So you have uncertainty about the variant and then uncertainty about what gene those variants are connected to. So how do we sort of try to approach that? So there's a few different types of approaches that one can take. One is you can use computational approaches, which takes advantage largely of the fact of two different things. One is that there's a lot now known about the genome, about where different variants lie, and do they lie in regions of open chromatin in particular cell types or things like that. Or you can look at patterns that you can see across loci on the assumption that if there's a particular functional annotation that is enriched consistently across multiple loci, that probably is telling you where the right variant is lying. You could also try to do an experimental system as long as you know it's relevant. And I'll tell you about one effort to do that for height as well. Or you can do more genetics, particularly if you can get rare coding variants. Those can sometimes help pinpoint what the right gene is, even if maybe the rare coding variant itself doesn't have a super great level of evidence. It can still sort of help shift your your probabilities of which gene is the right gene. So I'm going to tell you a little bit about computational approaches that we've taken. So one of the approaches that we developed is something called DEPICT, and we developed this in large part because when we first did our BMI GWAS, as I'll show you in a bit, the existing computational approaches really were not helpful in kind of pointing to the right genes. So this was a collaboration with a group from Groningen, particularly Luda Franca's group. And DEPICT basically took advantage of a huge amount of expression data and then a series of about 14,000 or so gene sets. And so the gene sets started out as sort of yes-no binary gene sets. So that's depicted by the matrix here on the right, where each of the rows is a gene, each of the columns is a gene set, and it's colored in red if somebody decided that gene was a member of that gene set. So I've actually drawn this matrix as much less sparse than it actually is because most gene sets just have a few genes, and actually many genes are not annotated as being members of any gene sets because most genes, nobody knows anything about them. So we wanted to have a method that could actually query any gene regardless of whether somebody knew anything about it or not. And so we took advantage of correlation across between genes in this sort of multidimensional expression space, and then used that to get a sort of semi-quantitative probabilistic estimate of how likely any given gene was to be a member of any given gene set. And so now we have a much richer matrix that we could then use to analyze the genome-wide association study data. And we took a similar approach also where instead of that matrix, you can use genes and expression in different tissues or cell types. And so you can get at, what cell types have genes enriched near associated variants and are also expressed in that cell type or that tissue. And so when you do that for height, you get things that make a lot of sense. You get chondrocytes as the leading cell type and mesenchymal stem cells as well. They're sort of precursor and cartilage as the right tissue. And that makes sense because the growth plate is where growing happens, and the cells that are doing the growing are the growth plate chondrocytes by proliferating, then hypertrophying, and then turning into bone. You also get gene sets that can come out of this approach. And so we got some relevant gene sets. And you can use this as well to try to guess at the right gene, although honestly, DEPICT is not quite as good at that as it is at the other two tasks. But even there, we had sort of a nice unintentional positive control where one of the genes that was the top predicted gene turned out after we'd made the prediction to be published as causing one of these monogenic disorders of skeletal growth. All right, so that's sort of like a nice just so story of like, oh, and we found a gene that turned out to be this disorder. But we wanted to not have to depend on that to know kind of whether DEPICT was any good or other methods that people had developed were any good. So we wanted to know, are we identifying the right genes or gene sets using these computational approaches? And the problem is that even for height, there's a lot of these OMIM genes. For a lot of other diseases, there are fewer of them, and it's still not clear that that's sort of the representative slice of biology. Genes that cause monogenic disorders have certain special properties that I can talk about if people are interested in, that may not reflect sort of what's being pointed at by the Genome-Wide Association study. So Rebecca Fine, when she was a graduate student in the lab, developed an approach called benchmarker, which is essentially kind of a leave one out approach, but we're taking advantage of the fact that the genome is very nicely partitioned in completely independent units of inheritance called chromosomes, and you can leave each chromosome out, predict which genes should be and gene sets should be relevant on the chromosome that you've left out, and then use the GWAS as your benchmarking metric to say, did you predict genes that have signals of association near them? And because the chromosomes are inherited independently, it's not circular. So we used benchmarker to compare 3 different approaches, DEPICT, and then 2 others, MAGMA and POPS, which we think actually wipes the floor with our approach. It's much better, developed by Hilary Finucane's lab. And we evaluated these using our height GWAS data. And what we found, in addition to POPS being better, at least for height, was that if you actually combined the methods, so look for genes that were prioritized by 2 or even all 3 methods, you actually did better using the metric that we used to benchmark, which is something from a method called LD score regression of per SNP heritability, or tau, for those who are interested. And so that's illustrated here, where the highest value for that metric was for these 356 genes in that intersection of all 3, although POPS and either of the other 2 methods actually does reasonably well and gives you more genes to look at. What was particularly striking was that if you look at the genes prioritized by those 3 methods, and they don't sort of know anything about sort of who is an OMIM gene, which genes are OMIM genes or not, but actually 16% of those genes were on our OMIM gene list, and a few others should have been on our OMIM gene list, but we can't count those after the fact. And that's compared to just a very few percent that are OMIM genes, just of genes randomly picked across the genome. All right, so, and we're still trying to, perfect how we use these computational approaches. The other approach that I'd like to tell you about for height is a relevant experimental system. This is work that's been led by Nora Renfall in the lab. It's been in collaboration with a computational postdoc who was in the lab pre-Anaka, and then Hank Cronenberg, who's at MGH and is a skeletal biology expert or extraordinaire expert. And so what Nora and Priya initially did is actually to look at the 3 zones of the chondrocyte. So there's illustrated here and then zoomed in here. There's the resting zone, or sometimes called round layer, the proliferative zone or flat layer, and then the hypertrophic zone. And basically differentiation kind of proceeds in this geographic pattern going through the growth plate. And so Nora also used as a different system a cell line from Vicki Rosen, which differentiates if you leave it in culture in the appropriate medium. And Nora was able to show that expression of late genes goes up as time goes on, suggesting there's at least a partial model of differentiation. So we refer to these as growth plate-like chondrocytes. And so what Nora and Priya did was to look to see was specificity of expression in any of those growth plate layers. Genes that had specificity for expression were those enriched in genes that were highlighted by the GWAS study. And so what she found here is that each of the 3 layers, round, flat, and hypertrophic, were actually all enriched. So that's sort of boxed in this yellow here. But then when she conditioned on specificity for the round layer, the signal for the other 2 layers went away. But when you condition on the other 2 layers, the signal for the round layer stayed. So specificity for the round layer being expressed in this early undifferentiated state was what was driving the signal. And it wasn't due to just recapitulating, finding these OMIM genes. These were a different independent set of genes. So that's sort of these conclusions. And then she saw similar results if you looked at genes that had specificity for being expressed at the early phase of that in vitro model. So now having sort of that in vitro model that has at least a couple of features that suggest that it might be a good model in some ways for growth plates in vivo, Nora then took a genome-wide perturbational approach. So she engineered Cas9 into these cells and then transfected them with a barcoded CRISPR library, and then allowed them to differentiate, looking for things that either where you had barcodes that were enriched in being positive for CD200, which is one of those genes that goes up late and happens to be a cell surface marker that has an antibody that you can use to flow sort. So either things that mature too quickly, or things that were CD200 low even after 15 days, so things that matured too slowly. And so here's the volcano plot that she got of those results. And you can see there's a bunch of genes that are either differentiate too early on the right or that differentiate too slowly on the left. And in addition to the sort of positive control of CD200, you can see a lot of interesting genes that come out. These include some genes that are known OMIM genes, and including some genes for overgrowth, interestingly, EED and EZH2, genes in clearly long-term known as being important regulators of the growth plate and in pathways, and excitingly, GWAS genes that had been prioritized by all three of those different computational methods. And the enrichment for GWAS got actually stronger as the sample size for the GWAS got bigger. So as you got better genetic information from the GWAS, the overlap got stronger. And I should say that Nora is pursuing some of the genes, some of the novel genes that have come up from this screen. So then you can also do more genetics for coding variants. So we did this as an initial attempt using something called an exome array, which is variants that are below 1% in frequency, but maybe not vanishingly rare. And through work that I sort of won't take you through the details of, but also this part of it was led by Rebecca Fine, collaborating with Giant. And so this is a blow up of this heat map here. Each column is a gene that has a coding variant that seems to be associated with height. And then using depict, these are the gene sets that are enriched in the GWAS data. And you can, if you sort of look at some of these names, these are highly relevant. And what you're looking for, is areas where the genes are strongly predicted to be members of those enriched gene sets. So you're looking for these clusters of red. And so this is a zoom in of one of those areas. And you can see that this annotation here is whether the gene was actually already known to be an OMIM gene. So you can see there's a huge enrichment in this little cluster. And so that suggests that the genes that weren't annotated as being OMIM genes were also likely to be highly relevant for skeletal biology. And again, we had another unintentional positive control where after Rebecca made this list, this figure, then there's a publication that SFRP4 was actually responsible for skeletal dysplasia. We also were able to validate some growth signaling pathways that had been proposed from work in mice and with some evidence in other genes from monogenic disorders, work led by Andrew Dauber. There's a gene called STC2, which inhibits a protease called in the PAPA and PAPA2, which cleaves the binding proteins, IGF binding proteins, which themselves sequester IGFs, which then lead to signaling that leads to growth. So if you follow all of those inhibitors of inhibitors of inhibitors, it predicts that if you knock down STC2 function, you should get more IGF signaling. And in fact, we found rare variants that in STC2 that increased height. And so the prediction was that those should actually decrease STC2 function. And we were able to collaborate with Klaus Oxvig, who in fact showed that these were hypomorphic alleles in STC2. So that predicts that if you knock down STC2 function, you should actually increase growth. All right, so that I've been talking almost exclusively about height, but we also have been studying obesity. And we've used a couple of different traits, body mass index, and then measures of central adiposity involving waist circumference and hip circumference. And so we had this sort of similar Manhattan plot. And this one, it was really very few obviously recognizable genes, although we have that parallel GWAS where we're sort of just analyzing the results now. and for body mass index. And at this stage, the sort of recognizable genes were sort of from a very familiar pathway downstream of leptin signaling in the brain, involving some of the genes shown here, plus actually an interesting gene, GIPR, which in light of sort of, incretin therapies being sort of the most effective therapies for obesity that we know of outside of bariatric surgery is kind of an interesting gene to have come up early. But most of the loci have no known connection to body weight regulation. When we use depict, we get sort of to my dismay, because I had not planned on being a neuroscientist, we get that the genes that are enriched in our genome-wide association study are somehow involved in the nervous system. I used to say brain, but the nervous system is in lots of different places, and we actually don't have strong evidence that this is central or peripheral or some sort of let's say gut-brain axis crosstalk. When we looked at the enriched gene sets, we saw some interesting things related to synaptic plasticity, and these are actually kind of meta gene sets, so clusters of gene sets that are all very similar to each other. And when we blow this one up, we see again things related to glutamate signaling and synaptic plasticity. And we got interested in this because these had been hypothesized to be sites of action of topiramate, which is kind of a messy drug originally developed for epilepsy, that has kind of a side effect almost of weight loss and anorexia, but with an unknown mechanism. So there's a group, we're collaborating with Dong Kong and a group in our lab of folks who are trying to figure out what the mechanism and the site of action of topiramate is, trying to use both in vivo in mice and neuronal cultures in vitro. When we do this for waist-hip ratio, we get a totally different set of tissues. So again, if I had to do it over again, I probably would have taken the lead on waist-hip ratio rather than BMI. But you get sort of adipocytes, which is a slightly more tractable system. And there's a group making some nice headway studying these. So when we did that same rare exome variant exome array approach for BMI, we got sort of about a dozen or less novel genes. One that I still find quite interesting is acetylcholinesterase. So you think about sort of cholinergic signaling. There's a well-known but toxic therapy for obesity called smoking with nicotine. But it certainly involves nicotinic receptors. And so one wonders whether this is sort of pointing to that same role. And then for waste, also, sort of a handful of variants, including some potentially interesting genes. And then when we do the pathway analysis, we get some other interesting genes like carbohydrate response element binding protein and others. All right, and so then I mentioned briefly sequencing. So what if you try to do really rare variants? So you can ask, we explained about 50% of height variability from common variants. Some of it's non-genetic. Is the rest of it rare variants, very rare variants? And at least coding variants is not sort of the full answer. Coding variants from biobank scale studies don't add much heritability, but they do point to genes. So this is the data from this cool browser called genebas.org, which took 400+ thousand exomes from the UK Biobank and looked for association with many, many phenotypes, including standing height. And the top one is actually one that we highlight from the GWAS studies as well, which is aggrecan, also an OMIM gene, sort of probably one of the more common unrecognized causes of short stature in heterozygous state. Same thing can be said about NPR2. And then SHOX, also well known. PAPA I'd mentioned already, LTBP2. Actually, this one was unfamiliar to me, and I looked and it wasn't on my OMIM list. So I said, oh, maybe this is new biology. So first place I went was to OMIM to see what was known about it. And in fact, it is an OMIM gene, just it appeared on, it was published again after I'd made the list. So yet another of these unintentional positive controls here. So this is an interesting gene as well. But the upshot of this is that if we want to get the same kind of results that we got from GWAS, we're going to need similar size samples or maybe even larger, but it will probably help to pinpoint at least some of the genes where there's a little bit more uncertainty from GWAS. Doing whole genome sequencing is also being done, but if we thought interpreting common non-coding variants was challenging, rare non-coding variants are going to be even more challenging, although if we can figure out how to do it right, probably informative, particularly for regulatory mechanisms. All right. So for the last little bit, and I put in a couple more stories here intentionally because I wasn't sure just how excited I was going to be and how fast I was going to talk. So I'm going to skip over one of them at least. But just to give an idea that genetics does do causal inference through something called Mendelian randomization, and it sort of helps untangle correlation causation. So I'll let you sort of look at this cartoon from XKCD. Yeah, so yeah, if you're giggling, you got it. But so genetics does offer a way of trying to disentangle that. And so I'm not going to actually tell you about this study, but it was led by Christina Astley, Jennifer Todd, in collaboration with David Ludwig, testing the sort of insulin secretion hypothesis for weight gain, which I have to say I came in as a huge skeptic of, although I know that there's, sort of certainly folks who advocate here as well. But in fact, it turned out that there was genetic evidence for at least some role of causality for insulin secretion after eating. I'm just going to skip through that. And just to say we're trying to do similar things with metabolite levels. So trying to understand, are there metabolites that might play a causal role in obesity? Because if you have the identity of the metabolite, that sort of gives you, again, a built-in physiological handle as to what the biochemical pathways that are relevant might be. And there are lots of genome-wide association studies of metabolites, which allow you to do this Mendelian randomization approach. And so we've studied, what metabolites we and others, many others now, are associated with obesity. And you find that a lot are. And when we tried the Mendelian randomization, we were initially very excited because it said that these metabolites were in a causal path, but then we did the opposite direction. We said, does obesity cause change in metabolite levels? And we got the answer yes as well. It's like sort of one of those magic 8 balls that kept giving you the same answer. So we've realized there's something more complicated going on. And it turns out there's ways you have to be careful about the ways you do Mendelian randomization. There's all sorts of violations of assumptions that can happen and something called pleiotropy. And so without going into details, there are methods to try to deal with this, and we're trying to use those methods and also extend it to metabolites that are unidentified. So you get thousands of unidentified signals when you do metabolite profiling. And so Yuhan Xu, using a method that's been published, has now a way of combining data for unidentified metabolites across cohorts, which you need to get the adequate power to do the studies we want to do. And we started to do those sorts of studies, and we actually get some interesting signals for a metabolite called alpha hydroxybutyrate, which coincidentally Vamsi Mutta, who I shared an office with, had also come across as a marker of not only insulin resistance, but hepatic reductive stress. So we're remain quite interested in that metabolite, and also did this with a bunch of unknown metabolites that might be interesting to follow up. And then finally, we're doing a study in collaboration again with Kara and David, as well as Clary from the Broad metabolomics platform and folks from the Channing, to look at metabolites that are altered in a randomized controlled trial of macronutrient intake, and then look to see, are those diet-associated metabolites causal for downstream outcomes? All right, so I did briefly mention predictive information, immediate impact. This is obviously true for when you sequence folks and you find Mendelian causes of short stature. And in fact, depending on how you ascertain populations, something like 10 to 20% of severe or syndromic short stature will actually have a Mendelian monogenic cause. Example, I mentioned ACAN. Again, often it was initially described with a very advanced bone age, so that people stop growing earlier and end up even shorter than you would expect. But actually, when you do sort of a genotype approach first, you find that there are a lot of people who don't have sort of the classical phenotype. And that's true for many, many different Mendelian disorders for reasons, again, I'd be happy to talk about. But I want to just briefly end with the possibility that maybe now that we have all this predictive information from common variants, maybe the polygenic risk scores can also provide some predictive information. And so we're actually now trying to test this. And again, I've been on the record early on as saying we will never get to the point where there's any clinically useful predictive information for height. So now maybe there will be. So we're looking now to see if you focus on the folks who are discordant from their actual height versus their genetically predicted height, are they more likely to have a monogenic cause? Are they more likely to have celiac disease or something else going on that's not just polygenic height? And we don't know the answer to that, but it's something we can now test, which is pretty exciting. All right, so I told you there are lots of associated loci for genome-wide association studies, and that even the common variants of small effect can be important, but it's hard to get from the association to the relevant effector gene in the biology. Talked a tiny bit about Mendelian randomization and sequencing, and the fact that we're testing polygenic predictors. So I've tried to acknowledge people on the way. There's a variety of people who have been involved in this work at Children's in the Broad over the years. And then some of the other folks that were involved in some of the other projects that I mentioned. And then I really want to highlight the work of the GIANT Consortium, which almost all of this either was or is the basis of the work that we're doing. It's just sort of this amazing large consortium that is tremendous fun and privilege to be able to help lead. So thanks very much. I'm happy to take questions.
How do you reconcile the differences that you see on the GWAS for BMI and the GWAS for waist circumference or waist hip ratio? I recognize that they represent different aspects of obesity or adiposity, but I would have thought that there would be a large degree of overlap.
Yeah, no, so I apologize for skipping over the fact I should have said it's waist hip ratio adjusted for BMI. So we actually adjust away all the sort of fat mass kind of things, and it's just distribution, or we try to make it a just a distribution phenotype. Yeah.
I'm one of the people who believes in the insulin secretion hypothesis.
Yes.
So let me put in a plug for that, number one. Number two, I was interested that you pulled out acetylcholinesterase, which may very well be related to that insulin secretion hypothesis because of the M3 receptor that sits on the beta cell per Matsuzaki in 2001. Have you taken a look to see whether or not acetylcholinesterase and M3 actually
We have not, and I have to confess, initially we sort of were like, oh, this is all brain, but there's, neuronal, or in this case, neuronal style genes, lots of other places. We have not looked. We have not sort of been the beta cell, so little of this has sort of pointed directly to the beta cell as sort of a group that we haven't sort of gone there, but it's an interesting thing. There are some folks doing beta cell work as part of the diabetes genetic stuff, and I'll pass that on to them. It's a good thought. Yeah.
It's probably coming out of left field from a different specialty, but I noticed in your dropout screen of the different stages of chondrocyte development that a whole bunch of cancer genes came up in the more differentiated stages, not being dropped out. EZH2 and SUS12 and all these cases. And so I'm wondering, first of all, do you think that height is related in any way to a more stem-like phenotype in these chondrocytes, and perhaps really getting crazy? Have you talked to anybody who studies osteosarcoma if they ask if any of these genes are mutated in case there's osteosarcoma, which absolutely just correlates with the
Yeah, it's a really good thought. We have not crossed our data set. I mean, there's tons of data sitting there in cancer atlases that we could easily cross it with, and it's a good idea to do that. I mean, certainly P53 isn't, gives us a little bang on the head to say, this is some of the same pathways. And I do think it's a stem cell type phenotype, especially the specificity for the early stages in the GWAS. And, it could be a lot of height is basically set very, very, very early on by how many stem cells you have and how good they were at replicating and continuing to replicate.
Maybe simplistic and naive, but in reference to your introductory comment about basketball players, I haven't seen a height cohort assembled of taller people, and could you learn something from just looking at the opposite side of the spectrum, and maybe tall people that are well above their height, parental height prediction, and things like that?
So at the moderately tall end, unlike at the moderately short end, you don't see sort of, at least on a population level, discrepancies from the common variants kind of do what they're supposed to do. But I think at the very tall end, it makes no sense. Like the model that I presented to you just does not fit. And there's something going on. I don't know if it's just a release of the brakes that happen and you just become a big outlier. But there shouldn't be like, people who are siblings and are both over 7 feet tall. It just shouldn't happen, and it does. And I have no idea why.
Yeah. This is the silly question of the morning. In non-human species, there seems to be a reasonably good correlation between body mass and longevity. Have you correlated either your height or your weight data with any, longevity in people is really hard to study.
Yeah. So we are in the middle of doing or I should say, that's the very broad the giant consortium led by Irini Maruli and others as part of our PHEWAS group. So PHEWAS, a phenotype-wide association study. So they've taken the polygenic risk scores for height, and they've looked across all the phenotypes, and they're basically done with their analysis, and I'm waiting to see kind of the initial draft of the paper. So I think the answer is there because there is some data about death in there as sort of a phenotype in the biobanks. But I'd have to make sure that they actually included it because you actually do have to take a little bit of an extra step. It's not an ICD code. You do actually need to specifically be looking for it. And it's a good thought, especially with all sort of the growth hormone deficiencies, especially in animals, et cetera, and, decreased signaling and increased life. So, yeah. Or 1, 2, maybe.
I have 2 questions slightly related. So first, this was incredibly impressive. 5 million of anything, of any trait, of course. Has to do with missing heritability. You didn't say it explicitly, but I'm sure you have thought about this. When the studies were smaller, about why we're only capturing a small percentage of the seems like if you'd be saying that with 5 million subjects, you're now explaining almost all really estimated twin studies or family correlations. Because others do this,
Yeah, we've estimated, we've captured the heritability that you can attribute to common variation. So using the methods that sort of try to partition heritability among common and rare. So we don't think there's missing heritability on the common side, but in some ways, you kind of expect that. I mean, those methods of partitioning explicitly get rid of all of the sources of missing heritability, I think. I don't know where the rest of the heritability is, the gap between 50% and 80%, whether it's interactions, rare variants, other things. Yeah.
So I think probably the last 50 years, the average height you talk about patient with achondroplasia, the little more, but that would be a major gene cause that much.
Yeah.
You didn't really talk about gene environment interaction or whatever, but are you thinking about that too? Because clearly there's a huge environmental effect going on here too, which could help elucidate the mechanisms for gene.
Yeah, so certainly heritability is much lower if you consider cohorts over time, right? It wouldn't be 80%; it would be like 40% or I don't know, something like that. Yeah, we've tried to look a little bit at gene environment interactions. Have not in earlier phases were not really that successful. Now there's better diet data in larger biobanks, and we could look at macronutrient intake and stuff like that. But I suspect that a lot of that sort of during childhood, where just again still the data are not that great. But there are some childhood cohorts, and I just don't think that, I'm not sure that anybody's really doing that, but they're probably, if somebody is, it would be the folks that are doing those childhood cohorts.
So do you think that difference over the last 100 years would be changed?
Yeah, I think so. I think it's nutrition. Yeah, I do, but I could be wrong.
So great work and great talk. Very few speakers can casually drop that they had a Nature paper yesterday.
Yeah.
So well done.
Well timed, yeah.
I have a question for you, and maybe it's better over drinks tonight, but I want to challenge the main premise about getting drug targets. It's going to work. And so decode started in 96, Amgen acquired them in 2012, Regeneron started with Geisinger and the others in 2014. If you look at the Amgen and Regeneron drug pipeline pages right now, there's not still a single drug in even phase 1 that came out of this. Now you could say PCSK9, but that started before all that population sequencing. So are the drugs really coming?
Yeah, there was just a Twitter thread about this, and I'm trying to remember whose page it was on that I just saw yesterday. It was actually in response to this paper coming out, and there was sort of that same question came out. There actually are a fair number of examples. I remember TYK is sort of an example that came out of this. BCL11A certainly for sickle cell came out of GWAS, and there was the list of about 10 or 15 that were on that, some of which I actually hadn't even heard of. So I think that, and it's still early days. If you think about this sort of generation time of, target validation to drug, or even entering really the drug pipeline in a way that maybe they would put it on their website. I think that I think it's still too early to sort of declare success or failure, but I think there are some early successes. So.
The bell is rung. Thank you for that excellent presentation, and we'll continue these really good conversations as we do a parade across the street and enjoy some good food and good discussions. So please come join us.